Welcome, everyone. I'm glad you were able to join us today for our webinar. My name is Michelle Jackson. I'm a product manager at Ambry Genetics and focus on hereditary breast, ovarian cancer, and endocrine products. Today, I will be talking to you about, specifically about hereditary breast and ovarian cancer and the changing landscape of that in particular. Just a very brief outline. All of us are very aware of hereditary breast and ovarian cancer, but I will give you just a very brief background. And then the main part of my session will focus on a literature review. I do have to say selective literature review, definitely not comprehensive. And then also I'll provide some of our internal Ambry data to go along with some of these topics that you're seeing listed underneath our literature review. And then once I've finished up my topic, we have a special guest from Malleola Foundation to talk about the second annual Battle for the Breast event and how your organization can become involved and potentially get genetic testing vouchers as a prize. So particularly for hereditary breast and ovarian cancer, historically HBOC has been synonymous with BRCA1 and BRCA2 gene mutations. However, the landscape of HBOC is now changing with the rapid identification of new genes and implementation of various multi-gene panels being offered. So it is increasingly difficult for clinicians and patients to keep up with this rapidly evolving field. So anything we can do to stay up with the current literature and learn more as we go is beneficial to everyone. So particularly for Ambry Genetics, about 50% of our mutation carriers are identified to have a BRCA1 or BRCA2 mutation. However, for another 12 to 30% of mutation carriers, they are identified to have mutations either on our BRCA plus or BRAF/NEXT panel. However, in addition, we have multiple other panels that include the BRCA genes, including GYN+, OvaNeXT, PinkNeXT, CancerNeXT, and CancerNeXT expanded. So clinicians can be ordering anywhere from 2 genes with just BRCA1 or 2, all the way up to 49 genes with the CancerNeXT expanded panel. Historical literature and various research studies have pretty much well defined the cancer risks, specifically for BRCA1 and BRCA2. These are the risks that we quote here at Ambry. I'm not going to belabor these. These are very well known to all clinicians at this point. There has been some debate and lots of literature, potentially with case reports or small studies looking at the cancer spectrum of BRCA outside of the risks for breast and ovarian cancer. This particular study by Mersch et al. Was published in Cancer this year in 2015, focusing on that topic as well. It was a single institution study, but was able to analyze over 1,000 individuals with BRCA1 or BRCA2 mutations, and just looked at those individuals themselves and their cancer history, not including any family history in this study. So as you can see from the table that I've listed here, definitely we still see the expected increased risk for female breast and ovarian cancer. However, among the total of about 30 cancer types that this group looked at, the only other cancers that they noted to have a significantly increased risk was in BRCA2 for pancreatic cancer and BRCA2 for prostate cancer as well. So the increased risk of cancers that I just showed you on the previous slide was confirmed by this study. They also did note a trend towards increased risks for melanoma and cervical cancer. However, other risks that had been potentially previously reported, such as colon cancer or uterine cancer, were not replicated in this study. So it seems that we're still sticking kind of with this core cancers for BRCA that that we've known about at this point, and I think the study assists with that as well. So if we start thinking about the up to 49 genes, at least that Ambry offers for hereditary cancer syndromes, and at least just focusing on the breast cancer genes and ovarian cancer genes, there's still quite a few. So this list just shows you the genes that are on our BRCA plus and breast next panels. And in the second column, you can see the associated cancers for each of the genes listed. Some of those are more well-defined than others. Some are still evolving as we test additional individuals and understand the phenotypes within families. In addition, for ovarian cancer risk, again, you can see all of the genes that are on GYN plus in the blue, and below that, all of the genes that are on our OvaNext panel, and then the associated cancers listed as well. So again, even just thinking about high and moderate risk genes for breast and ovarian cancer, there are many different cancers to consider or think about when evaluating families for hereditary breast and ovarian cancer, and not just thinking about BRCA1 and 2 any longer. So to focus on that a little bit more, and talking about some of the heterogeneity we're seeing in HBOC, this was a study done by Castera et al. Really the purpose of the study was to demonstrate the efficacy of next-generation sequencing. However, in addition, they identified that about 59% of their population did have a BRCA1 or BRCA2 gene mutation. The other 41% of individuals who tested positive had a mutation in another gene outside of BRCA1 or 2, and you can see that on the left-hand box of this slide. And I think this study really just highlights the heterogeneity of HBOC and helps us further understand that there are many other genes that can be responsible for hereditary breast and ovarian cancers. So to give you a little bit more specifically of Ambry data, one of our genetic counselors presented this poster at the National Consortium of Breast Centers in Las Vegas earlier this year, looking at BRCA testing versus second-line panel testing. And really the interesting points of this poster were that with the kind of lower suspicion cases, maybe where there's only one breast cancer, still young, or one case of ovarian cancer, the yield to find a mutation on a panel is actually higher than if you just look in BRCA1 or BRCA2. As you get more cases of breast cancers or ovarian cancers, the yield for BRCA1 and 2 definitely goes up, but you still have a significant number of individuals that are identified to have mutations on panels. So this, again, just supports that HBOC heterogeneity that I've been talking about. Another paper that's in press right now in the Annals of Surgical Oncology by Kapoor et al. Looked at this issue as well in a little bit of a different way. They did a retrospective chart review looking at just under 1,000 patients who underwent genetic testing. They broke their patients into two groups, patients who had only undergone BRCA1 or 2 genetic testing in the past, and then those individuals who had undergone multi-gene panel testing, and the panels varied from 5 to 43 cancer-related genes at that time. They found that their overall detection rate for BRCA mutations was about 3.8%, and didn't differ significantly between the groups of those individuals who only had BRCA 1 and 2 testing previously or were identified on a multi-gene panel. For individuals who had undergone a multi-gene panel and had identified mutations, they found that 7.4% of those individuals were identified using a multi-gene panel. The identification of the BRCA genes and non-BRCA genes wasn't significantly different within that group, but overall the detection rate was almost twice as high. And then looking at the variance of uncertain significance in this study, overall they found that the VUS rate for BRCA1 and 2 is about 4%. However, if you look at the VUS rate overall, obviously it's much less than BRCA1 and 2 only testing rather than on multi-gene panels. But as you add more genes and specific genes in particular, that VUS rate does go up. So this is a study where I just pulled out some of the data from Easton et al. In the New England Journal of Medicine in June of 2015. This was looking at the association of particular genes with breast cancer. This particular article did get quite a bit of press, and it's something we've looked into internally as well. So on the left-hand side, you're seeing the list of genes that they discussed in their article that they believe has an established association with hereditary breast cancer. On the right-hand side, you see the genes that are listed on our BreastNext panel in comparison to those genes particularly. The BreastNext panel was referenced in the paper as well, which is why I listed it here. So at Ambry, we do offer all of the genes listed on as the genes that they had established an association with breast cancer. Particularly on BreastNext, we had removed STK11. That's the gene that causes Peutz-Jeghers syndrome, for those of you who may not be aware. And that was because all of our internal data had shown us that individuals with STK11 mutations had other very defining features of Peutz-Jeghers syndrome, and we were not picking up any STK11 mutations on our breast panels specifically. We still do offer STK11 on other panels. So in addition to the genes that the article identified as having an established association with hereditary breast cancer, we do offer 7 additional genes on BreastNext specifically. And the authors did comment on these genes as well and felt that there wasn't maybe enough information or enough published information in terms of outside of maybe founder mutations or small studies that lent to this increased risk for breast cancer. So some of these genes will be highlighted in a few of the slides that I'll discuss today. So again, just going over some of our internal data and some of the cohorts that I thought might be interesting for this purpose. The top 5 mutations and accompanying VUS rates in either breast cancer diagnosed at or under 45 and breast cancer over 45 are seen in these 2 graphs. The common theme here is that the genes are the same, just potentially in different orders. So you see BRCA1, BRCA2, CHEK2, PAL2, and ATM, both in these groups. CHEK2 being the most common in both of these groups, and the ATM gene having the highest variant of uncertain significance. In another cohort, or another 2 cohorts from our internal data, looking at patients diagnosed with breast cancer only or ovarian cancer only. Again, same theme in the breast cancer only group with the 5 genes that I mentioned before. And then in the ovarian cancer group, I'm definitely seeing strong representation from BRCA1 and BRCA2, but also for RAD51C, BRIP1, and MSH6. So talking a little bit more about PAL2. So PAL2 is definitely one of the genes that has received a lot of publicity over the past year or so, particularly with this publication in the New England Journal of Medicine. So I'm not going to belabor this one too much, but just to put it in here kind of as a reference. And this paper really looked at PAL2 mutation carriers and the association of cancer risk with family history. So individuals with no family history had a breast cancer risk of about 33%, and those who had 2 or more first-degree relatives diagnosed with breast cancer at 50 years of age had a breast cancer risk of up to 58%. And then they also displayed this really nice chart that shows all of that data in terms of family history, groups located at the top, and interval years on the left-hand side. So this prompted us to take a look at some of our PAL2 data as well, and one of our genetic counselors presented this at the San Antonio Breast Cancer Symposium late last year. When we looked at our PAL2 data, we found about a 1% positive rate in our patient population. A very high percentage of those individuals with breast cancer with a mean age of diagnosis at 48 years of age. And then also a really high incidence of triple-negative breast cancers in this group as well, which was statistically significant when compared with non-mutation carriers. And almost an 8% of individuals with ovarian cancer in this group as well. We also did see trends for patients having a more family history of pancreatic cancer and prostate cancer, although those values did not reach statistical significance. Some of our data on RAD51C and D, this was presented by one of our genetic counselors at the National Society of Genetic Counselors meeting last year. And looking at some of our data from these 2 genes, at this point we were able to identify 33 mutation carriers in either RAD51C or D, in either families that had breast cancer only, ovarian cancer only, or breast and ovarian cancer. And interestingly enough, over 30% of the individuals with a mutation were identified in families with no reported cases of ovarian cancer. And just to display some of the family histories in our cohort, displayed on the right-hand side here. This is some very interesting data that we have on NF1. This was presented by one of our genetic counselors at NSGC last year as well, looking at mutation carriers and the clinical history of NF1. So it is a small subset of patients. There were 10 patients in this group. 60% of them did have a clinical diagnosis of NF1, but 40% of them did not. So focusing your attention on these top 4 lines here, 3 of those individuals had a history of breast cancer. 1 was diagnosed young. However, the other 2 were more average age of breast cancer diagnosis. No reported clinical diagnosis of NF1 or other features of NF1, and no family history of other features of NF1 outside of the potential breast cancer. However, all of these individuals were identified to have mutations in the NF1 gene. So I think this data brings up an interesting point of whether or not clinicians are not reporting this history when they send in patients for genetic testing, or are these other clinical features of NF1 not actually present in these individuals, and there may be some additional information to consider when thinking about NF1 and breast cancer. Some of our data on other cancers in ATM gene mutation carriers and their families. So this is data that was presented by one of our genetic counselors at ACMG earlier this year. And she was able to show that individuals who have ATM mutations were 2.3 times more likely to have a family history of pancreatic cancer, which was shown to be statistically significant. And that also ATM mutation carriers were more likely to have family histories of melanoma and brain tumors, although those weren't quite significant, but also just lending to more of a phenotype definition potentially for ATM mutation carriers. And then the last slide, kind of talking a little bit about HBOC heterogeneity, is the debate that we've all probably heard several times at this point about whether or not HBOC and Lynch syndrome have any overlap, or breast cancer might be associated with Lynch syndrome. So one of our genetic counselors presented this information at NSGC last year. We identified 112 patients with mismatch repair mutations for individuals who had undergone panel testing. As you can see from the pie chart, there was a various breakdown of whether individuals met HBOC and Lynch syndrome criteria, or only Lynch syndrome criteria, or neither, or just HBOC. And I think the interesting part of this is that 21% of those individuals only met HBOC criteria and not Lynch syndrome criteria, however, had mismatch repair mutations. And among that group, MSH6 mutations were most frequently mutated among breast cancers, ovarian cancers, or just individuals who met HBOC testing criteria. So I definitely think that this is still a topic up for debate, but hopefully additional research will be able to shed some light on that topic as well. Okay, so I'm going to move on and talk about some of the other topics I've listed for the webinar today, just briefly touching on genotype and phenotype correlations in BRCA mutation carriers. This is a topic that has been looked at for years and years, depending on how you kind of want to classify it. I'm not going to spend a lot of time on this information, but definitely you'll have access to these slides after the webinar, and are welcome to look at any of these studies or any other studies on these topics. This group in particular, many of the studies done in this particular topic are conducted by the SIMBA group, which is the Consortium of Investigators on Modifiers of BRCA 1 and 2, and looking at different ways to try to get at genotype-phenotype correlations. So many have looked at it by mutation location, specifically in cluster regions for breast or ovarian cancer, functional domains, whether or not mutations are located at the 5' or 3' end. Also by mutation type, which I think is a really interesting way to approach it, and hopefully will lend some good information from that as well. Also, histopathological predictors of breast tumors, those are pretty well established at this point. And then also by ethnicity. So particularly individuals in the Ashkenazi Jewish population and the founder mutations known in those individuals have been studied for years and years. However, there is some additional information coming out about mutations identified in African Americans, Hispanic individuals, whether or not they may have founder mutations, particularly the deletion of exons 9 through 12 has been reported a few times. Whether or not Hispanic individuals have a higher incidence of particular Ashkenazi Jewish founder mutations, and also looking at triple-negative breast cancer. And then also in the Asian population, and particularly PTEN somatic loss and triple-negative breast cancer as well. So too much data, obviously, to go over today, but definitely if this is a topic that you're interested in, there's a lot of data out there and a lot of studies to look up and read about. Another interesting topic is looking at SNPs as risk modifiers. So this was a study done by Couch et al. In 2013 to kind of address this particular topic, specifically in BRCA1 mutation carriers. So this study did genome-wide association studies and started with thousands of BRCA samples. However, narrowed it down to a little over 2,600 samples in affected and unaffected individuals with BRCA1 mutations. They also started with hundreds of thousands of SNPs and narrowed that down to the 17 most significant SNPs. Those SNPs were separated out into ones they felt were more significant for breast cancer and ones that were more significant for ovarian cancer separately. And from their study, they determined that breast and ovarian cancer risks can be modified by some of these SNPs. So particularly in the chart that you see here, the black lines in each chart denote the minimum and maximum risks for breast and ovarian cancer for BRCA1 mutation carriers. The blue line in the middle denotes the average risk. So almost 70% by age 80 in an individual for breast cancer, and probably roughly around 45% to age 80 for a person with the risk for ovarian cancer, which these numbers are what we're pretty used to seeing in clinic. And then also looking at the SNPs and how they can modify the risk. So the red lines denote the 5th and 95th percentiles, and then the green lines denote the 10th and 90th percentiles. And basically what they were able to show is by using these SNPs with someone who has a germline BRCA1 gene mutation, you are able to give them maybe somewhat more of a more personalized risk assessment number for breast and ovarian cancer based on this information. So I think this is a topic that is, still being studied and has a lot of interesting things coming out of it potentially. So just to kind of go back a little bit to address some of the ethnicity topics, I wanted to just display some of our internal data and show kind of the most frequent mutations we're seeing separated out by ethnicity. So the genes that are most frequently seen in these ethnicities are located at the top. The ethnicities that we separated out are listed on the left-hand side. And as you can see, most of the mutations are concentrated in the 5 most common genes that I mentioned before: BRCA1, BRCA2, PAL2, CHEK2, and ATM. However, there are some really interesting outliers, so particularly in individuals of Middle Eastern ancestry. We're seeing some RAD50 mutations. Individuals of Hispanic ancestry, we're seeing ATM and MSH2 mutations. And then in Ashkenazi Jewish individuals, interestingly enough, we're seeing some NF1 mutations. Then also just to show you a very small snippet of our DUS data by ethnicity. So I've only focused on the panels that really are significant for hereditary breast and ovarian cancer. However, one of our genetic counselors, Laura Panos, has submitted an abstract to ASHAG and will detail this topic in much more detail at that meeting. So just to kind of summarize some of this information, you can see our overall VUS rate broken out by panel. And then if you look at individuals who are Caucasian, which is the largest sample size we receive, they do contain some of the lowest numbers outside of the Ashkenazi Jewish individuals for BRCA1 and 2. And the African American and Asian populations definitely have the highest VUS rate per their ethnicity and by panel as well. So switching gears a little bit and talking about triple-negative breast cancer for a while. The study was done by Couch et al. Earlier this year in 2015 in the Journal of Clinical Oncology. They looked at 17 breast cancer susceptibility genes and were able to detect 14.6% of those individuals to have deleterious mutations in one of the genes they identified. 11.2% of those were in either the BRCA1 or BRCA2 genes, which is not surprising, but there's still a good portion, about 3.7%, were identified in 15 other genes, PAL2 being the highest of those outside of BRCA1 or 2. They also identified an earlier age of diagnosis and higher grade tumors in triple-negative breast cancer patients with mutations when compared to those without mutations. So the summary from the study basically said that for triple-negative breast cancer patients, regardless of age of diagnosis or family history, should be considered for germline genetic testing of at least BRCA1 and 2. Currently, there are NCCN guidelines that state for individuals diagnosed at or under the age of 60 should be offered genetic testing. And so I think there was some interest in this specific recommendation based on their data, and Dr. Domchek out of the University of Pennsylvania submitted an editorial on this particular article, just reiterating that this study, their particular data, only showed that the frequency of triple-negative breast cancer patients over the age of 60 was only about 1.4%. And so currently our threshold to offer genetic testing should be at 5 to 10%. And so that those individuals, there wouldn't be enough in that group really to consider additional genetic testing, and really that this data reinforces the appropriateness of the NCCN guidelines, and that, we should remain offering genetic testing to triple-negative breast cancer patients at or under the age of 60. So some interesting commentary there. Additionally, with this study, this is just a chart where they showed all of the different genes that they examined in the triple-negative breast cancer patients. Obviously, you can see the number of mutations identified in BRCA1 and 2 are the highest, and then PAL2 being the third highest, and the distribution of the rest. Interesting enough, there were no CDH1 mutations, no CHEK2 mutations, and no STK11 mutations in this group. So to kind of give you the Ambry side of this, we looked at our triple-negative breast cancer patients identified with mutations identified on panels, and show the mutation frequency breakdown here. So BRCA1, the most common, PAL2 is actually second in our cohort, and then BRCA2 follows, and then the other genes you can see listed here, and then their frequencies. Of note, about 85% of the triple-negative breast cancer patients that had mutations tested at Ambry were identified either on BRCA plus or BRCA-NET, which isn't necessarily a surprise, but just kind of noting that these individuals, all of these individuals, didn't necessarily have the largest panel we offer or anything like that. So some genes may not have been analyzed more often than others in this particular population. In our group, we did find about 11.9% of triple-negative breast cancer patients tested positive, and the age of diagnosis was roughly 4 years younger in those who had a mutation than those that did not. We're still analyzing some of the family history data to take a look at that as well. So to move on to talk about male breast cancer, this is a topic that I'm personally interested in, but it's very rare, and so it is quite understudied, and the genetics of male breast cancer in general remains poorly described. I think panels could potentially help us with some of this information as we go forward and counsel patients, male breast cancer patients in particular. Right now, many of the studies are being done in specific ethnic populations. They'll have publications on male breast cancer patients, Italian male breast cancer patients, or male breast cancer patients with Jewish ancestry and things like that. And so it's still very limited. There's very small numbers looking at some of this information. There was a particular study done this year that reported a new large deletion in male breast cancer patient that had a very extensive family history of breast cancer. So not a surprise that it was a BRCA mutation, but just describing a new large deletion that had been identified. In addition, there are a few studies looking at somatic sequencing in male breast cancer, which I think is a really interesting topic as well. There's also an international male breast cancer program that presented some of its findings at the San Antonio Breast Cancer Symposium last year, and they presented on some of their survival data for male breast cancer, but aim to include some genetic information, family history, and other topics as they go forward. And the whole point of this program is really to try to gather more data collectively about male breast cancer to raise the numbers and the power of some of these studies to learn more about male breast cancer in general, which I think is a fantastic initiative. Some of our internal data on male breast cancer. On the left-hand side here, you can see the most common mutations and their accompanying BUS rates for our male breast cancer patients. So BRCA2, not a surprise, is the most common mutation we identify. But then CHEK2, PAL2, MSH2, which I'm not sure what to make of at this point, and then ATM as well. The mutation spectrum we also includes NF1, MRE11A, BRCA1, and TP53. Some of this information and much more additional information actually was submitted to the San Antonio Breast Cancer Symposium for this year by one of our genetic counselors, and additional information will be presented there. Just looking at some of this data that we currently have, the average age of diagnosis for men who tested positive wasn't significantly different from those who tested negative, except when you looked at the CHEK2 founder mutation. The age of diagnosis, or the average age of diagnosis in those individuals, was about 46%, which was significantly lower than those men who were otherwise positive for a different gene mutation. Switch gears again a little bit and talk about BRCA Pro for a few minutes. This was a common tool I used in clinic, and I think many people use in clinic. And looking at some of the studies that have been done recently on this particular topic, there's a study published in the Journal of Clinical Oncology this year looking at ovarian cancer. So it was a multi-institutional study that looked at just under 600 patients diagnosed with invasive epithelial ovarian cancers and calculated BRCA Pro scores for these individuals. This study really found that BRCA Pro significantly underestimated the chance of a BRCA1 or 2 mutation if the BRCA Pro score was under 40%, and that underestimate was most evident, particularly in high-grade serous ovarian cancers. Another study published this year in The Oncologist looked at men being tested for various reasons. This was a single institution study. Some men had personal histories of either breast, pancreatic, prostate, or other primary cancers, or no cancer at all, and presented for testing either based on a known familial mutation, a personal history of cancer, or a family history of cancer. And for men, the BRCA Pro model actually showed that it had reasonable sensitivity, specificity, positive predictive value, negative predictive value for men. So not so great still with ovarian cancer, but okay for men in general. And then most recently, in Cancer Informatics this year, there was a publication on some BRCA Pro enhancements that have been made. So I thought you all might find this useful and interesting. So some of the enhancements include that BRCA Pro can now calculate age-specific risks for contralateral breast cancer. It's able to deal with missing current ages or ages of diagnosis, and beefed up some of their tumor marker information as well, and being able to calculate the relationships between those markers. In addition, family members undergoing oophorectomy, bilateral mastectomy, and or female or male mastectomy, and then also some information on race and ethnicity as part of their enhancements as well. Okay, so that's all of the information to kind of talk about kind of more breast cancer-focused topics. I'm going to take a few minutes and just touch on the hereditary ovarian cancer part of it, and looking at the information kind of beyond BRCA there. There's a study published this year in Gynecologic Oncology looking at this specific topic. They examined 911 patients who were either diagnosed with ovarian, fallopian tube, primary peritoneal, or breast cancer. all who had previously tested negative for BRCA1 and 2 gene mutations. Interestingly enough, they did not identify any FTK11 or MSH2 mutations in this group. Overall, this population had 7.4% mutation or variant-likely pathogenic rate. The US rate was about 28.4%, and then the rest were negative for any gene mutations. Specifically looking at the ovarian cancer cohort in this group, BRCA1 mutations were identified most commonly in ovarian cancer patients who tested positive. Of those ovarian cancer patients who have Lynch syndrome, about 75% of those were identified in the MSH6 gene. One individual had 2 mutations in ATM and MSH6. And for the ovarian cancer group, they did not identify any mutations in TP53, CDH1, or BARD1. And you can kind of see the breakdown of the mutations they did identify over here on the left. Looking at a little bit more of that data, particularly for the breast and ovarian cancer group, you can see the most common mutation identified was CHEK2, and then the breakdown of the other mutations identified. There was no difference in average age of onset for breast or ovarian cancer in this group. So that really concludes the data that I'll present. I also just wanted to take this opportunity to make you all aware of some upcoming oncology posters and presentations we have at upcoming meetings, particularly ASHG, NSGC, and San Antonio Breast Cancer Symposium. We will wait until the end for any specific questions, but I will now turn it over to Jacqueline Talpesh, who is our community outreach manager and specifically works with our Malleola Foundation, and we'll talk to you more about the Battle for the Breast event that's coming up and how you can become involved. I think I will hand it over to Jackie. Hopefully she has privileges now.
Yes. Hi, everybody. Good morning. So my name is Jacqueline Talpesh, and I am the Community Outreach Manager at the Malleola Foundation, and I'm going to talk to you about the upcoming Battle for the Breast. So Battle for the Breast, what it is in summary, it's an online surfing competition. We did our first one last year, and through Surfline, Malleola, Ambry Genetics, the genetic and surfing communities, and social media, it expanded to approximately 10 million viewers. So this is great publicity for your clinic, for your foundation, and everybody else that's involved. So this event is hosted by the Malleola Foundation, and it's presented by Ambry Genetics, and it is powered by Surfline. And for those of you that are unaware, Surfline is the main surfing hub for the entire surfing community as a whole. So right here, this is an image from last year's Battle for the Breast on the left. What we have is 16 of the top female surfers in the world, and they're going to be gathering video clips of their best surfing moves. And the world's top 16 female surfers will be paired with 16 clinics and their partnering foundations to compete for up to $50,000 in hereditary breast cancer testing vouchers, adding up to a total donation of $125,000 from Ambry Genetics. So what I'm going to be talking to you about today is how to get involved, how to sign your clinic up, and see if you're eligible, and just give you guys an idea of how it went down last year. So this is an image from surfline.com last year as well. So this is an image from round 1, which starts at the kickoff for Breast Cancer Awareness Month, October 1st. So in mid-September, we will be doing a drawing where we pair each professional surfer up with one of the clinics. So we'll have lots of you guys signing up for a chance to be paired up for one of our surfers. And it goes live September 11th. It's going to be during the Hurley Surf Pro happening in Trestles. And it goes live on Surfline. So again, round 1 you can kind of see as a visual. Each girl is paired up there. So how it works is there's going to be a round of video clips. So each video clip, you get to watch each clip. Each surfer will post their video. So you watch 2 clips back to back and vote for your favorite one out of those 2. And then you go to the next 2 and the next 2 and the next 2. So then it goes from round 1 to the quarterfinals. So first round after the first week, 8 of the clinics and surfers will be eliminated, and we go to these groups here. And then from the quarterfinals, we go to the semifinals, where we've got 2 clips of our professional surfers each. And then from there, we go to the finals. And in the final round, it's always really fun. Everyone gets lots of publicity from it. All the girls post on it, and each of these girls have millions of followers on their own. And then last year, winner was Bethany Hamilton. You guys might recognize her from various movies and whatnot. She's very well known in the surfing community. And she placed first in the online voting competition. And Bethany represented BreastLink and Cancer Research Collaboration. And that is our Executive Director, Hans Hagen, presenting the checks to both Cancer Research Collaboration and BreastLink. So the donation is made on behalf of Ambry Genetics to the foundations, to the foundations. So what happens is Cancer Research Collaboration will receive the donation in forms of test requisition forms. So you will receive, if you participate, a pink test requisition form, and from there you will be eligible, and Ambry Genetics handles all of the information. And a lot of you are probably wondering how to sign up for this, and luckily for you, we actually go live today, which is really exciting. So today on the actual website, you can go online to Malleola or Ambry, and it will link you to our page where you can sign up and register. So just another idea of how much you are eligible to win for. This is a list of our top placements. So first place was Bethany Hamilton, and she won her clinic $50,000 of hereditary testing vouchers. Second place was Tatiana Weston Webb. And then third place was third and fourth is what Ty won it, since they're both eliminated in that last final round, and it was Sally Fitzgibbons and Laura Enever. And for this year's girls, we have, we're going to go to this slide first. This is a list of all of our professional surfers that participated last year, along with the clinics that they represented. So if you want to look at this list, you can kind of get an idea of whether or not your clinic is eligible. You can kind of see there. I'm also going to go into more detail on that because you want to make sure you're connected to a 501c3 foundation. This is a list of all of the professional surfers that will be competing in this year's Battle for the Breast. And if any of you guys are surfing fans, most of these girls, if not all, are competing at the U.S. Open of Surfing now happening at Huntington Beach. So it's always really cool. If you guys want to go check out Surfline right now, you can even check out these girls in advance, and they are currently gathering their clips and their videos that they're going to be using to post for this competition. So step 1, registering. This is where you guys really want to pay attention to make sure that you guys get all signed up and everything. So register the foundation associated with your hospital or clinic for a chance to win up to $50,000 in hereditary breast cancer genetic testing vouchers. Ambry Genetics is giving away a total of $125,000 in free testing vouchers. So submissions as of now, they're due September 11, 2015, and as I stated, it does go live today. So you can go online right now even, and you can check it out on our website. It's going to be malleola.org. And just to make sure that you are eligible, this is where the foundation registration is. You can look up your tax ID number or your 501c3 for your foundation right here. And you just type it into the website, and that way when it pops up, it will tell you the name of the foundation, make sure it's all correct, and then it will also link you to a registration page where you enter your contact information and whatnot. Step 2, the lottery. On September 11th, during the Hurley Pro at Trestles Beach, a lottery will be held to the pair of 16 hospitals and clinics and their associated foundations with the top 16 women's professional surf athletes. So at this lottery, we want you guys to know that this is a random drawing. So once you guys all submit your information, we're going to have the girls be drawing the actual names. We're going to do, like last year we did ping pong balls, and we had all the numbers written on ping pong balls, and we're going to film it. It goes live, so again, it's big publicity for all of the clinics involved, and it's completely random, and then we will be drawing 16 total, and each of these girls will be paired up automatically with their associated clinic. So step 3, the goal of this contest is to spread awareness about the importance of early detection in breast cancer. During the month of October, which is Breast Cancer Awareness Month, you can visit surfline.com and vote for the surfer or clinic of your choice. At the end of the voting period, Ambry Genetics will be donating complimentary genetic testing vouchers to the pairs that receive the most votes. So So we're just asking you all to join the battle, and you guys can go check us out on Facebook or Instagram. We've got our hashtags on there as well. Again, Malleola Foundation and Ambry Genetics, Surfline, all of the surfers will be posting about this, and it gets pretty crazy during the month of October. Everyone gets really enthusiastic about it. So definitely reach out to us if you guys have any questions. If you guys want to contact us to see if you're eligible, just go to the website. You guys are free to email me. And, yeah, let me know if you guys have any questions. And we're wrapping up. So thank you again, Michelle, for the introduction. And if you guys have any questions, we're going to go ahead and open that up, and you guys can type it into the Q&A on the side there.
Thanks, Jackie. And to all the participants, if there are any questions, please feel free to type into the Q&A like Jackie just mentioned. I do see one question listed at this point that says, will a list of these articles be provided after the presentation? The slides and the webinar itself will be made available on the Ambry website within 1 to 2 weeks after today. And so you can definitely get all of the information there. I would be happy to send a list of the articles to the particular person that asked for that or anyone else that wants them as well. Are there any other questions that anybody else has? Okay, so we just received another question that says, as new data comes out, how often do you reevaluate what genes are on the panel? I think at least from my perspective, we are reevaluating genes on a daily basis. As new data comes out, we look at it, we, examine the data, we analyze it, that's in their particular articles, and try to figure out, what that means, how it could benefit patients, if there's enough data on that particular gene at that point to make it clinically available. So I think I would say we do this on a, daily basis, really. We're obviously not always updating our panels that frequently, but I think, some of the data that comes out, we need, more data before we can really justify putting that on a panel or offering it clinically. I think from a clinician's perspective, if there are genes that you want to see or, feel like there is a lot of data out there and want them to be offered, definitely email us and, or talk to your representative in the field, and they will definitely relay that message back to us, and we can take that into further consideration along with the literature that's out there. There's another question that came in that says, will there be any follow-up additional info for the MSH2 mutation in male breast cancer? That's a very good question. So 2 of our genetic counselors have submitted an abstract to San Antonio Breast Cancer Symposium on male breast cancer. I'm sure that they will address that information there, but I think male breast cancer in general is something that we want to look at additionally and, focus on a little bit more. So if there is additional information that we have, we'll definitely let you all know. Any other questions anyone else has? Okay, I don't see anything else coming in right now. Definitely, if you do have other questions, feel free to email me directly or, talk to your field representatives and have them submit your question, and I would be happy to respond to you at any time. If there is additional questions that come in, I would be happy to respond to those individuals directly as well. So thank you all so much for joining us for the webinar today, and I hope you enjoyed it, and have a great day.